I'd like to thank the organizing committee for invitation. Charcot-Marie-Tooth neuropathies are one of the most common inherited neurologic conditions affecting 1 in 2,500 individuals in the United States. Both children and adults are affected, causing motor and sensory deficit, dysfunction, pain, and need of ambulatory aids. Severe childhood cases may end up requiring ventilatory aid or they may become wheelchair dependent. Classic CMT clinical features would include slowly progressive weakness and atrophy of distal limb muscles, which are more pronounced in lower extremities, as one would see in a length-dependent distal axonal disease. With progression of the disease process, atrophy and weakness of the intrinsic hand muscles and forearm muscles would ensue. CMT1A is the most common inherited neuropathy and results from a 1.5 megabase DNA fragment, duplication of this DNA fragment on chromosome 17. This DNA fragment encompasses a gene that encodes peripheral myelin protein 22. So individuals with this condition ends up having 3 copies of this normal gene, and the protein is very dose sensitive, so the overexpression results in the dysfunction compromised in the Schwann cells. PMP22 is an integral membrane glycoprotein of 22 kilodalton. It's the hydrophobic protein, has a tetrastrand structure. It constitutes about 2 to 5% of the proteins in the compacted myelin. So it's a structural protein, and also it's believed to have a function in cell cycle. In fact, initially it was discovered in fibroblast cultures as growth arrest stimulating factor GAS3. This particular mutation, duplication, occurs in 70% of all CMT1A patients. Therefore, the most common mutation in the CMT neuropathies. I like to emphasize 2 main features of CMT1A. One, there's a preferential distal axonal loss, which is a very important feature of CMT neuropathies that directly correlates with the clinical disability. Secondly, there's also an impaired axonal regeneration that contributes to reduced numbers of myelinated fibers in distal nerve, supported by studies in xenografts from CMT1A patients showing a delay in the onset of myelination and impaired nerve regeneration, and in the Trembler-J mice, a naturally occurring animal model for CMT. I like to use this little cartoon to point out the unique place of CMT in terms of classification of neuropathies. The traditional teaching calls for a classification according to the anatomical site of involvement. Where do we put CMT in this scheme? As far as genotype is concerned, it is a myelin disease, myelopathy, because it carries the PMP22 point mutation, which is a primary Schwann cell genetic defect. Electrophysiology shows typical uniform slow conduction velocities, therefore it is a myelin disease. But the clinical phenotype is typical of distal length-dependent axonal disease, so-called dying back neuropathy. This point is very important because the decreased number of axons, our distal axonal loss, directly correlates with the clinical severity, and this axonal disease results from impaired Schwann cell axon interactions. For the axon, especially the distal axon, to stay viable depends on healthy functioning Schwann cells around them, and primary Schwann cell genetic defects have profound alterations in the axonal cytoskeleton and membranous organelle movement, leading to degeneration of the distal axons and clinical disability. So somehow, if we can bypass the primary Schwann cell genetic defect, perhaps we can induce axonal growth, promote axonal growth, and that would reach the target and bypass the clinical disability. It is now well established that Schwann cells are absolutely essential and play a crucial role in support of axonal regeneration and associated myelination. We hypothesize that primary Schwann cell genetic defects interfere Schwann cell ability to differentiate into a competent state to support axonal growth and myelination. And Schwann cell survival factors such as NT3 could overcome these deficiencies. NT3 is an important autocrine factor supporting Schwann cell survival and differentiation in the absence of axons. During embryonal development, Schwann cell precursor survival solely depends on neuroregulin coming from axonal contact. Embryonal life E18 and on forward in the adult nerve, the Schwann cells can survive without axonal influence by establishing this Schwann cell autocrine survival loop. And IGF, PDGF, beta beta, and NT3 are the main components of this autocrine survival. Priming Schwann cells with NT3 might be beneficial to nerve regeneration and associated myelination as first step in the treatment of CMP neuropathies with primary Schwann cell genetic defects. In our previous studies, we showed that a subcuticular injection on NT3 showed efficacy in animal models and in patients with CMT1A. In an earlier study, the subcuticular NT3 treatment in nude mice harboring CMT1A xenografts showed improved axonal growth and associated myelination. Here, this cross-section of the xenograft 2 months after NT3 treatment shows a very robust axonal sprouting compared to the untreated xenograft, and the BDNF treatment showed no benefit whatsoever, and it is also not a member of the Schwann cell survival loop. In a study 2005, we also showed that the Trembler-J mice and the patients with CMT1A showed enhanced nerve regeneration. In these studies, this was a double-blind, placebo-controlled, small pilot clinical trial that included 8 CMT1A patients. Sural nerve biopsies, fascicular type sural nerve biopsies were performed before and after treatment to assess the effect of NT3 on nerve regeneration. And the clinical outcome was neuropathy impairment score. I like to have you to notice these clusters of multiple thinly myelinated fibers within these regeneration units. This typical feature occurred in post-treatment sural nerve biopsies. And these tiny thinly myelinated fibers are the result of the treatment, and we quantitated these myelinated fibers within the regeneration units. And this slide briefly summarizes our results. In sural nerves, a biological effect of NT3 on nerve fiber regeneration was unequivocally established by a significant increase in the mean number of small myelinated fibers within the regeneration units. The graph shows the least square means and standard errors of mean myelinated fiber per regeneration unit in a log scale at baseline and 24 weeks. As seen here, the treatment, NT2 treatment, resulted in a highly significant increase in the number of regenerating myelinated fibers within the regeneration units that I showed you in the previous slide. The clinical outcome showed significant improvements in total neuropathy impairment score and sensory gradation scale in the distal limbs of pin, vibration, and cold temperature modalities. And NT3 was well tolerated with no significant adverse events. Although these studies have provided proof of principle data showing that NT3 provides nerve regeneration in animal models as well as in the patients with CNP1A, the short half-life of NT3, which is only 1.3 minutes following intravenous administration in rats, would pose practical difficulty in its clinical application. Gene therapy approach, on the other hand, would offer the potential for sustained NT3 delivery through secretion by muscle cells. Continuous low circulating levels can produce a therapeutic effect with a minimal risk of toxicity, and systemic production of NT3 by gene therapy would be more convenient and cost-effective than repeated injections of the purified peptide. The premise of our studies is that intramuscular injection of NT3, AAV1 NT3, will provide histopathological, electrophysiological, and functional improvements in CNP1A. For these studies, we chose Trembler-J mice model, which carries a point mutation in the same gene that is duplicated in CNP1A. As seen here in these pictures, the Trembler mice nerves show severe hypomyelination, decreased axon caliber, increased neurofilament packing density, and increased hypophosphorylated state of the neurofilaments and impaired regeneration. In earlier studies, we first show that the intramuscular injection of AAV1 NT3 in Trembler mice resulted in therapeutic and persistent levels of serum NT3. The slide here shows the results at 23 weeks post-injection of AAV1 NT3, and the bars are the NT3 levels in individual animals. There is a significant and variable NT3 results compared to the control values. And the treatment also resulted in both regenerating, following a crash of the sciatic nerve, in regenerating as well as the intact sciatic nerves, a significantly increased number of myelinated fibers. and this was associated with improved hind limb grip strength. Furthermore, in the intact sciatic nerves from the trembler mice, myelin thickness increased, indicating that the treatment improves hypomyelination, a myelination state of the trembler nerves. As illustrated in this figure, we have apparent increase in the myelinated in the myelin thickness of the sciatic nerve axons, and the G ratio determinations show this quantitatively. There is a shift to the smaller numbers, meaning thicker myelin in the treated group compared to the untreated group. G ratio reflects the myelin thickness. It's the ratio of the axon diameter to fiber diameter. Our main objective is to carry the AAV1-NT3 gene therapy to a clinical trial. In order to achieve that, we should conduct a translational study in NT3 mice that will enable the use of compound muscle action of potential amplitude, CMAP amplitude, is a reliable marker for functional improvement in CMT neuropathy. This is our experimental design to achieve that. The vector that we used has the human NT3 coding sequence under CMB promoter. The animals received a 2 × 10⁻11 viral particle of the vector into the quadriceps muscle. Sciatic nerve conduction studies were carried on at baseline, 20 and 40 weeks post-injection of the virus. The functional studies included bilateral and ipsilateral hind grip strength and rotor performances done weekly. And we also carried out some correlative morphological studies. These include muscle fiber size determinations in the ipsilateral limb that we measure the conduction velocity of that limb and in an anterior and posterior compartment muscles. And we also assess the axonal filament cytoskeleton by ultrastructural morphometric studies and Western blot analysis. These are the representative traces of sciatic motor nerve conduction from wild type and Trembler J mice at baseline and endpoint 40 weeks following AAV1-NT3 gene therapy. Please note the scale, the height of this square is here 2 millivolt, here half a millivolt, and 1 millivolt at the endpoint. The CMAP amplitude of the mutant mice is significantly smaller compared to the wild type, and the morphometry of the CMAP amplitude is polyphasic and prolonged, and there is also a prolonged distal latency in the trembler mice. At endpoint 40 weeks later, we see in the same animal a significant increase in the CMAP amplitude, which went from 0.33 to 0.08 millivolt. And keep in mind that this is the neurotrophic effect of the circulating NT3 because we injected the NT3 into the quadriceps muscle and we're getting the nerve conduction velocity determinations from the sciatic nerve. Among all the parameters that come from sciatic nerve conduction studies, CMAP amplitude is most often used to assess the state of the motor nerves because it is proportional to the number of conducting motor axons in the nerve, and it's a reflection of the number of motor nerves exactly is in contact with the muscle. Here's the Cmap and conduction velocity in the sciatic nerve at 20 weeks post-therapy. There is a 37% increase in the Cmap amplitude in the NT3 group compared to the PBS injected group. All other parameters were insignificant. The bilateral hind limb grip strength and the left hind limb grip strength also showed a significant improvement compared to the PBS received trembler mice at 20 weeks. And this slide shows the relationship between hind limb grip strength in electrophysiology. The 27% increase in the Cmac amplitude is corresponding to a 29% greater grip strength in the AA1-NT3 treated animals. At this point in the study, we used some of these animals to assess the neurofilament cytoskeleton to assess the effects of the AA1-NT3 injection on the neurofilament cytoskeleton. Keep in mind that trembler mice shows an increased neurofilament packing density compared to the wild type, and this is associated with an increased hypophosphorylation state of the neurofilament. I should say here that the NFH subunit of neurofilaments, they form the side arms coming from the neurofilament structure, and they're heavily phosphorylated, and it is believed that the negative charges in the side arms actually is the force keeping the normal neurofilament spacing in the wild type. The NT3 treatment resulted in normalization of the, or toward normalization of neurofilament packing density, as seen here. The number of neurofilaments per a unit hexagonal area was decreased compared to the untreated group, and this is illustrated here in the middle panel at the ultrastructural level. Our Western blot studies complement the ultrastructural changes that we have seen in our morphometric data that we generated. At 20 weeks, the phosphorylated NFH content was significantly greater in the peripheral nerves, sciatic and spinal nerve end groups compared to the PBS treated group, corresponding to a less densely packed neurofilament spacing. Here's the electrophysiologic data at 40 weeks post-injection. Now we have an 84% greater Cmac amplitude in the AAV1-NT3 group compared to the PBS group, which is highly significant. And there's also a mild increase of the conduction velocity, which is significant but modest. Moreover, compared to the baseline, the treated group, A1NT3 group, showed a 28% increase of CMAP amplitude, which went up to 52% and became statistically significant at 40 weeks. During the same period, the PBS group showed a gradual decline, correlating with the natural progression of the neuropathic process. We continue to collect functional data from these animals between 20 and 40 weeks. As seen here, the NT3-treated group performed much better in the rotor-rod testing. And here's the composite histograms of the myelinated fiber size distribution at 40 weeks. Now we have a highly significant increase in the mean fiber diameter in the NT3-treated group compared to the PBS group. And this increase is more prominent for fibers less than 5 micron diameter. Here's the histogram showing the muscle fiber size distribution from an anterior compartment muscle and a posterior compartment muscle from a lower limb. Over 2,000 muscle fiber diameters from 3 animals in each group were recorded, and the data shows that there is a significant increase in the mean muscle fiber diameter as histologic evidence of improved nerve regeneration into the muscle. So in conclusion, Trembler mice receiving IM injection of AAV1-NT3 show improvements in the hind limb grip strength and CMAP amplitude. Accompanying this, there are improvements in the myelinated fiber density, myelin thickness, axonal cytoskeletal properties, as well as the muscle fiber diameter. The CMAP amplitude can be used as surrogate for clinical functional improvement, which has direct relevance to future clinical trials as a reliable outcome measure. These data provide proof of principle evidence for considering gene transfer of AAV1-NT3 in CMT neuropathies as well as other nerve disease with impaired regeneration. I now like to acknowledge my wonderful colleagues for their unending support and help for this study, and a group of extremely talented technicians that we have in our center and the funding institution. Thank you.